presentation view. Great, thank you.
Okay. All right. So, following, Helen's excellent talk, I think I'm going to move this topic to a little bit to the clinical side. And, I'm a, basic scientist, researcher, and, as a training. And very luckily nowadays, I can collaborate with a lot of clinicians and surgeons in Taipei, and so that we can identify new genes involved in not only brain development, but also brain developmental disorders. And this is just a very brief introduction of, what can go wrong in our during our brain development. And, for example, this is a normal brain where you see a lot of really nice organized gyri and sulci, and the cortex and the white matter is really, really nicely structured. However, in some cases, when the brain development has some errors, you can have a smaller brain called microcephaly, or a smooth brain called lissencephaly. So I was really, really interested in these kind of brain developmental disorders as a PhD student, and now as a PI. These patients also suffer from a lot of problems clinically. For example, epilepsy, developmental delay, learning disability, and autism. And I'm sure I don't have to go through this slide. I just like to emphasize that during brain development, there is a really, really important process called neuronal migration, where these neurons have to leave their germinal zone where they were born, and then migrate for long distance to populate the developing cortex. And over last almost two decades now, we did develop methods to visualize this process. For example, how these radioglial cells divide and how these neurons migrate. And this is the ORG cell that Julian mentioned, that we identify in the mouse. And also we can see, look at the subcellular structures, for example, the mitotic spindles, the centrosomes, and then etc. So using this powerful tool, we really can dissect the entire process of neurogenesis and also neuronal migration during cortical development. After coming back to Taiwan, I was really thinking, what I should do as a career development. And I was thinking maybe I can, I've been working on mice for many years, but I think that mice is interesting, but I think if my research can translate to human patients, that would be wonderful. So I started to collaborate with clinicians in Taiwan, and start to identify maybe new questions from the clinical side. And at that time, my neighbor, the Taipei Veteran General Hospital, very close to Yangming University, they have a population of patients who has this so-called focal cortical dysplasia, or FCD. Instead of having this entire brain have malfunctions and malformation, these patients only had a small portion of the brain that has some defects. For example, in this patient, it has some so-called transmental sign, where you see the white matter and the cortex has blurred boundary. And the scientists have been studying this problem, and at that time, we thought that maybe this FCD is caused by some somatic mutations in these neural stem cells. Remember that a neural stem cell can produce a lot of neurons that migrate to the cortex. If there is a mutation during development in the neural stem cells, they will produce a population of neurons that has some mutations in their genome. These are so-called somatic mutations in these cells. And this population can actually has some dysfunction, and they emit abnormal signals. So usually that these patients would have a focal epilepsy where they started this abnormal electric signal and spread out to the entire brain. So I was thinking, what I can do to these patients, and this process can happen in the human. Can it happen also in the mouse brain, and can I mimic this process using the animal models? So one day I was thinking about this question, and it came to me that when I was a college student, I read a book called A Feeling for the Organism. Maybe some of you have read this book, and this book is about Barbara McClintock, who discovered transposons back in the 30s in the maize, and actually. This has become a really nice tool to mutagenize the genome or somatic cells and also germline cells, so that people can use this to identify genes important for example cancers. So I was thinking, I've learned this in vitro electroporation technique, and I can introduce genes into the brain. Can I use this as a tool to mutagenize these neurostem cells? So this is the idea where I can use in vitro electroporation to introduce these transposons. And when the transposon hit a gene that is not important for neuronal migration, differentiation, or proliferation, then these cells can actually migrate to the cortex normally. If the transposon hit an important gene for this process, then these cells may get arrested in the deeper layers. Okay, so we just got the transposons from our collaborators, and as a control, you can see when we electroporate GFP, and 2 weeks later, you see most of the cells have migrated to the cortex. When we electroporate this transposon without the enzyme transposase, then these cells still can migrate to the cortex normally. But when we add these transposase cDNA together with the transposon, then actually you see a layer of normal neurons in the cortex, but you see an additional layer of neurons arrested in the white matter, which is really interesting. So we can take out this piece of tissue and then run through a series of PCR, so-called Spinkerette PCR. Then we can identify the neighboring sequence where the transposons is inserted to the genome, so that we can know exactly where the transposon is inserted. So using this technique, we actually identify many genes that may be involved in cortical development. So we identify about right now 30-something potential genes, and actually many of the genes have been implicated in some human brain developmental diseases. For example, this CDON, this STX1B, etc. And that gives us the confidence that maybe our genetic screening protocol actually worked. So these are some others. We even identify microcephaly gene and etc. But we need really to go back to the mouse and see whether taking out these genes will cause brain developmental diseases. And we also did some in silico experiments. We actually run into get the software called IPA, and we look at what the common pathway or common functions of these 33 genes in animals, and then we actually see that these development keeps popping up. You can see our development of the embryos, cellular development, nervous system development, and it is not associated with other, for example, cardiovascular development disease or cell morphology or connective tissues. So our screening paradigm really can identify potentially new genes involved in this developmental process. So then we went back again to knockdown each gene one by one and look at whether these genes is really involved in this process. So again, we use shRNA to knock down these genes, and as a control, you can see most of the cells have migrated to the cortex. And when we knock down these few genes, you can see a lot of cells are still arrested in the deeper layers. We also can rescue this using a cDNA to rescue these migration defects. So we tested almost all of these potential candidates. We found that at least about 2/3 of these genes, when we knock them down, you get an arrest or at least some delay in neurons redistributed to the cortex. This black bar is the cells still in the VZ and SVZ, and the gray bar is in the cortex. And in the normal condition, most of them have reached the cortex. But when we knock down many of these, you see a lot of cells are in the deeper layers. Okay, so as I said, I really want to go back to the patients and see if these mutations can also be identified in these FCD patients. So we collaborate with the clinicians and surgeons here, and most of these patients, there is only one way to treat the epilepsy for these patients, that is this epilepsy surgery, where the surgeons take out a piece of this lesion, okay, until the epilepsy comes down. Then we can take out the piece of tissue and then look at the pathology and identify what type of focal cortical dysplasia these patients are. And also we can take out the fresh brain tissue and then extract their DNA and compare the potential mutations in this piece of tissue with their peripheral blood. And we run through a pipeline of next generation sequencing, and so that we can read the genome for hundreds of times. Then we can identify these somatic mutations in this piece of tissue. Using this method, and we specifically looked at these 33 genes that we identified using our mouse genetic screen, we identified at least 3 of them overlap with the patients that in our population. That's DSCAM, contacting 1, and CEP152. And we also did a knockdown of these genes, and we found indeed this gene knockdown can delay neuronal migration in this the mouse brain, and we can also use CRISPR Cas9 system to show that disruption of these genes can really delay cells in the deeper layers. So this paper has been published in 2018 in Nature Communications. To summarize, we identified many genes using a really interesting transposon-based somatic mutagenesis in the mouse. We identify many genes that's potentially involved in this cortical development process. And some of them have been known before, for example, this CEP protein, DMD, and MACF1, etc. But we also identify many other genes that has not been implicated in this process. So I think that we can actually study each, one by one, what's the function of each genes during this process. So I think this paper really provide a very powerful tool to identify new genes in cortical development. And we also went back, we also have recruited a lot of patients with malformations in cortical development. And we actually identified some other interesting genes. And one of them is really interesting, that is involved in the centrosome. So during neuronal migration, we also identified that the function of interesting protein, the lysencephaly-1 protein, that is involved in the centrosome movement during neuronal migration. So using a live cell imaging in the brain slice, you can see central movement is really, really interesting. It would lead the way to move and then move into the leading process, and then the nucleus would actually follow. And if you knock down LIS1, you can see that the central movement is really disrupted. And really interestingly, very recently, we in a Taiwanese patient, we identify a new gene that is appearing in a patient with lissencephaly. And using next generation sequencing, we identify this new protein CEP851 in these patients. And in collaboration with a lot of geneticists, including Australian scientists, we actually found many other patients who has these mutations in this CEP851 gene, and this is MRI, a 3D MRI imaging. Is actually most of these patients has posterior dominant lissencephaly, which means that the posterior part of the brain is more severe than the frontal cortex. And of course, the first thing we would really want to look at is whether these mutations also has some neuronal migration defects. So we knocked down this CEP851 gene in the mouse cortex, and we found indeed it actually affects neuronal migration, but it did not affect neuronal differentiation, because when we stain the cells with the neuronal marker, you see there are actually pretty much the same percentage of cells have differentiated into neurons. And we also look at the localization of this CEP851, and you can see that because it's a CEP protein, that indicates that this protein is probably a centrosome protein. So we express these proteins in the centrosome and use a super resolution microscope. We can see this is a flag tag for the CEP851. We can see it's actually in the proximal end of these centrioles, and very close to the PCM, the Paris centriolar matrix. Okay, so this is also true in neurons. You can see it is also very localized to the centrosome in the migrating neurons. And using a 3D structure illumination microscopy, or 3D SIM, this is actually unpublished, we can look into more details of the localization of this CEP851. And you can actually see it forms a ring. It forms a ring in the centrioles. And this is really, really interesting. And we are still working to identify its real function in the centrosome. And in the same time, we also identify another patient who has also lissencephaly and also some muscle weakness. And this is also an interesting case, because after next generation sequencing, we identify a gene, so-called BicD2, or the Drosophila ortholog gene by Caldo. And we found this mutation in this patient, but when we look back to the literature, we found that this gene mutation, BicD2, actually is associated with a type of spinal muscular atrophy. So it's not associated with brain developmental disorder. But when we look at the mutation site, this is really interesting, because in all of the other mutations associated with SMA, they are all missense mutations. That means that only an amino acid was substituted with another amino acid. But in our case, we actually identify this a truncation mutation in the C terminus. So this is very, very different. And we really want to know whether this truncation would cause brain developmental disorder. So we express this mutation in the cortex using in vitro electroporation. So as you can see, when we express this truncated BicD2, you can see a very severe delay in neuronal migration. But when we express the SMA-associated mutants, you can see most of the cells still can migrate to the cortex. So we then look at whether the centrosome movement is also affected. And we found that this is really interesting because the BicD2, when we express the truncated form, the centrosome movement is really not affected. It actually moved to the leading process. However, the nucleus cannot move. So this is very different from what we saw in the LIS1 knockdown cells. Then we go back to the literature and found that actually BicD2 may be associated with the nuclear envelope. So when we express it, you can see very nice nuclear envelope staining. And also it recruits the dynein to the nuclear envelope. But when we express this truncated mutation, you see the protein was spread out to the cytoplasm, and also the dynein no longer be recruited to the nuclear envelope. And when we express these SMA-associated mutants, you can see the nuclear envelope localization is not affected. We can also do this using biochemical assay. We can use extraction of different cell compartments. We can identify this mutation is actually more associated with the cytoplasm, but not the nuclear fraction. Okay, so later on, we also identify is an interacting protein. We actually found that BicD2 interact with a nuclear protein called Nesperin-2. Okay, and then it recruits the dynein dinetic complex. And our model is now that this BicD2 it actually needs to recruit dynein onto the nuclear membrane so that it can transport, the dynein can transport the nucleus forward along the microtubule cytoskeleton. If you have a mutant BicD2, although it can interact with dynein, but it cannot interact with Nesperin-2. So actually it has acting as a dominant negative effects for this gene. Okay, so this is our current model. We use the shRNA and also expression of these proteins in the cortical in mouse cortex. And we saw that LIS1 dynein actually is acting to move the centrosome forward, and the BicD2 and dynein needs to move the nucleus forward. We are still not clear what the function of CEP85L is during this process. So we are still studying this. Okay, I think this is my last slide, and a lot of work, a lot of people, my students contribute to this work, and now we are hoping that we can understand with not only the genes but also the mechanisms how these genes and proteins would affect neuronal development and